this kind of interdisciplinary aspect of SGD research and integrating the biological aspect and really trying to come up with a cohesive story of how that plays together and is impacted by submarine groundwater discharge has been something that I've been working on for a few years. And it's a field that I'm very excited about because I think there's a lot of potential for new ideas, new growth, and new findings. So my talk today isn't necessarily going to be about my specific studies, although I will talk about a few of them and what I think the key findings were from those studies, but more about like the history of submarine groundwater discharge, biological research, highlighting some other really good studies that have been done by scientists that very much impressed me in their methodology and their findings, and kind of like setting the frame for like what we can all do to develop and integrate a little bit more the biological side of submarine groundwater discharge. So as always, I think it's good in these, especially when you're in the SGD community, to give our definition of submarine groundwater discharge since it can vary study to study. And today I'm talking about everything. So this will be fresh groundwater, it can be brackish groundwater, it can be recirculated seawater that is getting enriched in the coastal aquifer or in the subterranean estuary and then discharging back out to the ocean. And when I talk about SGD biology research, I'm really talking about studies that have actually documented a response to submarine groundwater discharge or attempted to document that response. So that could be as simple as they took chlorophyll samples along with their radon survey, or they did an incubation experiment. They looked for changes in biodiversity or some sort of organismal abundance. Not enough to say that, you know, SGD provides so much nitrogen to this bay per day, and therefore it can cause, you know, X some amount of primary productivity. I've done that too, you know, in multiple papers as well, but that doesn't necessarily document a response.
Hey, Alanna.
Oh, you guys can't see it.
Exactly. We don't see your screen yet.
Good thing I'm on my second slide. You would think I'd be more practiced at this after doing this for an entire year, every day. Okay.
No, but it was so clear we didn't need the screen yet, but soon we will probably.
Yeah, soon you will. You didn't need it yet. Okay, still on the first slide. So as I'm saying, like the studies I'm going to be specifically talking about today work to actually document a response. And I will say that even if they don't find a response, which oftentimes is the case in SGD research, like, or they find a mixed response, maybe they don't find a correlation with chlorophyll and radon or whatever they were hoping for, the more of that data that we get, the better our understanding is going to be for the future. So even if we don't find some significant finding about how groundwater discharge at this location influenced the biological aspect of the ecosystem, just having that data, I think, is really going to set us as a community a lot further into the future and more advanced into making the outside world, outside of us, you know, scientists, understand and care about submarine groundwater discharge. I'll be talking about that a little bit as well. So first I want to talk about this figure that we just made that came out in the paper led by Isaac Santos and Nils and many of us other SGD researchers around here as well, where we made this table just to look at all the SGD research that we could find where there was a documented response to nutrients in submarine groundwater discharge. So this didn't take into account other factors like pH or salinity, just nutrient discharge. So just nitrogen and phosphorus for the most part, since that's what most people study. And what was the response of the biology of that system to SGD? And we can see that it's not always the same. We can have increases in organismal abundance. We can also have decreases. just at the species scale. At the community scale, we have only really 3 studies that have looked at species richness. So this is the number of species in that area, or the number of genera in that area where that's being exposed to SGD. We have 1 study showing an increase, 1 study showing a decrease, and 1 study showing unclear or mixed results. So this is why I don't think it's enough to kind of say, SGD provides so much nitrogen or so much phosphorus, and that can lead to so much primary productivity in an ecosystem, because it's not necessarily always the case. And I'm going to talk a little bit about that in some example of other studies, what other studies have found. So first, I want to do a little poll about SGD research to get an understanding from you guys of what your barriers are to integrating more of the biological aspects into your study. I'm going to talk a little bit about the history of SGD biological research, a few of my studies, and I think what the major take-home points have been, a few other great studies that I think some of which have been very overlooked in terms of what they've been able to tell us about SGD biological interactions. We'll talk about the database that I'm working on building for NILS, which is supposed to actually make this a lot easier for the average SGD scientist who is not a specialist in biology to find relevant papers for them. And then also a list of recommended readings or papers that I thought were particularly good that you can continue to explore if you haven't read them after my talk today. So first, I'm going to ask you to go to socrative.com. And then you should click login, which should be in the upper right-hand corner, student login. The room name is It Is the Way. And then you should have a question pop up. There's going to be 3 questions. And I will paste the questions. So after we get a good chunk of responses from this one, I'll move it on to the next one so I can share the results with you guys. So once you've answered the question, just keep the window open.
It's cool. I'm learning interactive teaching techniques. Thank you.
Yes, working at a primarily undergraduate institution. This is the type of stuff I get to pick up and learn about. So it looks like our most common response is most people have published about 1 to 5 studies. Of course, there's still filtering, but it seems to be about 40% of the folks here today. Next most common is we have about 30% of people here are just working on their first. So congratulations, good luck. And then a fewer who have worked in 5 to 10 and then beyond 10. So if you didn't get a chance to respond, don't worry, I'll have more questions coming up. I'm gonna move this forward onto the next one, which is a little bit more specific to today's talk. So how many SGD studies have you completed that specifically assessed a biological impact of SGD? So did you measure chlorophyll, do a survey, counted cells? Okay, so for this question, really the bulk of the responses, like 90%, are split evenly between 1 to 5 studies and 0 studies. So I think this is actually good news, because for the 1 to 5 studies, it means that you have some experience on which to expand. And for the 0 studies, it gives you lots of new areas. Maybe you'll find someone, a collaborator in your own institution where you can do this potentially in the future. And I'm going to talk about what I think are some like easy ways to integrate more biological documentation into our research. And then last question. When you write a manuscript and you haven't necessarily done biological, any documentation of biological response, what's the biggest barrier to integrating previous research about biological impacts into your discussion? So you have a bay, you've calculated the nitrogen flux of that bay, you want to add some information in the discussion about what the biological impacts might be. What do you think are the barriers to you? And I'm just going to leave this one open, so you feel free to continue filling this out, because I realize this one might take a little bit longer because it's actually writing an open response. It may even take you a while to think about what that might be, but we will come back to this. All right. So next I want to talk a little bit about the history of SGD biology research. So like, where has it come from? What's happened? And what does that landscape kind of look like? It's been assessing the impacts of biology in terms of how submarine groundwater discharge impacts biology. It's had a lot more attention in the last few years. I've seen just an explosion of studies of different scientists coming up with very unique ways to document that response. But it's actually been going on for a while. Like, many of these studies occurred before I was born. So like 1967, we had our first study. This was not an SGD specific study. It was actually looking at coral reefs in Florida and assessing like what was causing the deterioration of many of the coral reefs. And they found that it was actually lowered salinity due to submarine springs near the reefs that was actually causing that damage. So all the way back to 1967. We had very few studies, and a lot of these early studies were like that. They weren't SGD specific studies. And a lot of these studies in general are not like that. So they're in journals where SGD is kind of mentioned offhand as, oh, this is what's causing this problem. You know, like we went to go, they found the problem, damage to a coral reef, for example, that's a very common one, and then went to go look for what the sources of the problem might be for that, and they found out it was groundwater. So they didn't go there wanting to initially study SGD, which is kind of like the opposite way that many of us approach SGD studies. And in the last few years, we've had a great deal, as I said, of SGD studies, looking at just lots of different techniques, lots of different locations. So there is a great, actually really good body of research. I think at this point, we're almost up to like 100 studies specifically looking at documenting that effect. We've had a little bit of a drop in 2020, I think, probably due to things outside of our control. Notably, the pandemic obviously slows some of these things down, or 2021, and we're just getting started. But already I've seen like some good papers come out about SGD research, SGD biology research. What's been studied? What have these studies focused on? A lot of them have been on microbes, so just like group like phytoplankton and bacteria. So whether it's single-celled algae or bacteria all together here, often studied just because that's often what we care about. A lot of times the justification for like we're studying SGD in this area is because we think it's going to cause some sort of algal bloom. So, or contribute to primary productivity. A lot of the primary productivity obviously comes from these very small microscopic organisms. Surprisingly, there's actually been a lot of work on macrophytes. So these are like plant-like organisms. So whether they are a large algae like a kelp, well, there actually haven't been any SGD studies on kelp so far. So area of inquiry if you live in an area with kelp, but things like sea lettuce or large marine plants. Chordate animals, so these are anything with a spine. These are mostly fish, have been very, very recent, and they've been really only come out of a couple of different labs. And I have to say I've been very impressed because that's a very hard leap, I think, to be able to document the response so far up the food chain, all the way up to fish. And so that's about 7%. Non-chordate animals is about 23%. So this is animals without a spine. And really, the bulk of it is corals. There has been a lot of research on corals with respect to SGD. And most of that is because it's coral researchers going out studying corals, finding some sort of negative effect, like maybe there's no reef building corals in that area. You need certain corals get restarted. Other corals come in and build off of those corals to expand the reef. So like that effect of having a reduced population of reef building corals, or ocean acidification that causes ocean damage to corals, or any number of things, too much nutrient loading, which causes other competitors such as turf algae to come overtake the corals. A lot of times these studies are done by coral researchers that happen to be in areas where there's a lot of SGD or a spring that's impacting them. They're not SGD specific studies. And then also ocean metabolism, which I would call like maybe more of these chemical indicators that not necessarily a more biologically focused would take, but like maybe someone who's more chemically focused, such as accounting for respiration, calcification, things like that. There's only been a relatively few on that, which I think would also be another, because that gives you a view of like the whole ecosystem as a net, what's going on. I think that's a great area that should be looked into more in the future. What's been studied a lot? Chlorophyll. Chlorophyll's easy. It takes like 2 days to measure. You only need like 100 mils of water per water sample. You just scoop it up in a cup, put a lid on it, throw it in the cooler until you get back to the lab. You only need a fluorometer, which are not, you know, when you're talking about like Radex and Rad7s, a fluorometer is not that expensive. Lots of marine institutions, which many SGD researchers are situated at, already have a fluorometer. They can go in a shared lab, or you know, someone that they can borrow for a couple of hours, because it really doesn't take that long to measure chlorophyll. And I think that's the first step of entry for a lot of people into SGD biology research. And I think we actually need more chlorophyll data. Diatoms, unsurprisingly, SGD is very rich often in nitrate and in silicate. Diatoms need both of those in order to grow. And actually diatoms are the only phytoplankton that's really large enough to take advantage of nitrate. Nitrate takes a lot of physiology to break down into a usable form. It takes a lot of energy. Diatoms are huge in terms of the phytoplankton world. So they have the space, they have the ability to take up nitrate and to break it down into other forms. Comparatively, if we look at areas where SGD is rich in ammonium or dissolved organic nitrogen, there we might see more of an impact on dinoflagellates. Dinoflagellates are a lot smaller. They cannot take up nitrate very well or use it, but they're very good at using these other forms of nitrogen. So depending on what's in the groundwater, we may see a response either from diatoms or dinoflagellates, but these have both been the focus of many studies. Like I said, corals, most of the invertebrate studies have been focused on corals. Even just looking at like histories of corals recording groundwater discharge at a site over decades, or over, you know, I think I've seen like up to like 90 years. They'll use corals for that, which can also document response. And then also ulva species. So this is basically sea lettuce. I've got a little photo for you down here in the corner. There have been a lot of studies of sea lettuce. So this is a genera. So any of the species within that genera, For a few reasons, it's ubiquitous amongst coastlines. It's everywhere. So it's actually would also be a great species if we get enough data that we can actually really compare across different sites. It's very easy to identify. You don't need a microscope for it, like you might for phytoplankton. It's very easy to sample. You just go like cut a piece of it off. And it's because it's so big, it's easy to do other analyses on it, such as like nitrogen isotope analyses to really see if the sea lettuce is taking the nitrogen up from the groundwater or from another source. It's a very common way of studying sea lettuce, or NDP ratios. It's another common thing that we look at with sea lettuce. So just because it's so big, it's easy to take a lot of sample and to identify it and do those analyses. What's been understudied? Surprisingly, bivalves. So this is like, there's been, I think, one study that has done it well with mussels and one study that's done it well with oysters, which is kind of surprising because there's been so many SGD studies that look at submarine groundwater discharge and the nutrients discharged through it in areas where there's like oyster reefs and mussel beds. But that link hasn't been very solidly made yet. And I think there's so many ways that we could do this, right? We could do a radon survey and see if radon is higher in areas where we have oyster beds potentially. Or for all the remote sensing folks out there, you can look for the ways that they identify SGD hotspots, such as changes in salinity or changes in temperature. You could see, does that coincide with oyster beds? And there is a good reason that mussels and oysters would be very impacted by SGD. I think that's also been rather overlooked. Yes, having a lot of primary productivity from nutrients in the water would be good for mussels and oysters, but really, mussels and oysters, all of their predators tend to only do well in high salinity waters. So such as whelks, for example, which predate oysters and mussels, cannot live in brackish water. So a area where you have oyster beds and much lower salinity could be an indicator that oysters are thriving there as a result of the SGD just because of the lower salinity. But we haven't really had a lot of studies that really look at the direct link between mussels and oysters and SGD. We've had more about fish, which fish they don't even stay in the same spot, right? They're much harder to study. Oysters and mussels are also really cool in that, much like corals, they create bands. So if you have a little bit more of a paleo mindset, you can look at historically maybe SGD discharge by sampling those bands and looking at different isotopic tracers. So there's a lot of potential, I think, with mussels and oysters. Fecal indicator bacteria. There's been about 6 or so studies, including one I think published this year, on how submarine groundwater discharge impacts fecal indicator bacteria. Unfortunately, I think there's just so many unanswered questions in this realm. So despite the fact that we have 6 studies, you think we have a pretty good understanding of it. But it seems that there'll be areas where there's a lot of very high concentrations of fecal indicator bacteria or abundances in the groundwater and very low in the ocean. And then other areas where groundwater does seem to effectively transmit fecal indicator bacteria to the ocean, or where it supplies something that the bacteria need to grow. We just don't have enough data to have an overarching theory and I think understanding of how SGD impacts fecal indicator bacteria well. Fauna in the sediment. So animals that live in the sediment. Most of the studies look at, once the SGD gets into the water column, how does it impact the biology there? But there's so much going on, even in just the top couple layers of sediment, biology-wise, that can impact submarine groundwater discharge. So this can be male fauna, which are very, very small animals that live in between the grains of the sediment. So that might be things like nematodes, or larger marine worms could also live in the sediment. And just to give you an idea of how important this is and how understudied it is, there's one study which looked at SGD impacts on marine flatworms, which only live in like really the upper centimeters of the sediment. And these marine flatworms have algae inside of them that's symbiotic and uptakes nitrogen. What they found is that these marine flatworms are basically intercepting a lot of the nitrogen before the groundwater discharge to the ocean. So if you're using that end member of nitrogen concentration, I think they're specifically looking at nitrate, and multiplying that times the discharge to get how the flux of nitrogen to the ocean, and you're not accounting for that, all the nitrogen that's being lost in that last couple, inches or last couple centimeters, then you're vastly overestimating the nitrogen flux to the ocean. And then also whole ecosystem studies. So studies that look at the response of the entire ecosystem, which are a lot harder. They require a lot more skills than, say, just taking chlorophyll or just looking for sea lettuce, which is really easy to identify. But looking at diversity, richness, and evenness, these are ecological indicators that have been around in ecology since the 60s. There's only been a couple of studies that have looked at things like diversity and richness. But integrating more of that, I think, is going to be really important. But those have been very impactful studies, I would also argue. And other things like net processes, like there's one study, I think, in Hawaii that showed that, okay, SGD increased primary productivity and oxygen content, but it increased respiration more. So it actually drew down the oxygen content of the water. That's an important finding, calcification, things like that, or other net biological processes. Okay, let's take a look at, we're going to look at two of my studies. The first one is this one, which I think is the one that's a little bit more well known, which is nutrient loading, specifically in Monterey Bay, California, where there are recurrent harmful algal blooms, specifically Pseudonychia, which is a harmful algae. It's a diatom, and it produces, when it's stressed, so only when it's stressed does it produce domoic acid. And domoic acid causes domoic acid poisoning, which can impact marine life, birds, whales, dolphins, but also humans, because if it accumulates in shellfish and humans eat that, then it's a neurotoxin, actually. And like Alfred, I don't know if you guys have seen the movie The Birds by Alfred Hitchcock, but he was actually inspired to make that movie based off of these recurrent algal blooms in Monterey Bay, because he was visiting Monterey in the Santa Cruz area, and he saw like all these birds that were flying into houses and into cars because they had so much domoic acid poisoning in their brains that they couldn't fly straight. So we did two parts of the study. The first part was we did 2 incubations, so basically grew phytoplankton and fed them various nutrient treatments, and then also local groundwater to see their response. And then a field study to quantify the nutrient flux of SGD to the bay. I'm just going to focus in on these 2 incubations today because I think it's the more interesting part of the study and maybe the more important impacts in terms of research for others. So we had various treatments. We did 2 incubations. We learned from the first and the second one based off of that. These 2 figures on the left are from the first incubation, and the 2 images on the right are from the second incubation. This is the total cell count, so the total number of cells that we counted under the microscope for each treatment. So we had the initial amount of cells, control, which was after 3 days with no additions. To some of the treatments, we added 10% groundwater, 20% groundwater, soluble reactive phosphorus, nitrate, ammonium phosphate, and urea. Saw a big response in groundwater in the first study, or in the first incubation, but we were unclear as to what in groundwater was actually causing that. Because if you look at this, you can see that nitrate, the total cell counts, did not go up nearly as much as like the groundwater count. So we're wondering what did we miss in our experiment, which is why we actually went back and did the second incubation, which had lower concentrations of groundwater to try to be more realistic. And we also added a nitrate plus silica treatment. Groundwater in that area, groundwater in many areas, is very high in concentration of silica just because of the rock-water interactions. The groundwater is just dissolving silica and discharging into the ocean. You can see here that the nitrate and silica treatment produced just as many cells as the highest groundwater treatments, very similar across the groundwater treatments. And if you look at the type of phytoplankton we're looking at, I mean, we put these into very broad categories. So the only genera we looked at was Pseudonychia, because that was this what we cared about because we were specifically looking at those blooms. We also looked at centric diatoms, which form circles, and pennate diatoms, which form chains, and then also dinoflagellates, because those are often talked about in the SGD research as well. You can see from both these figures on the bottom that it's mostly diatoms that responded to the SGD or to the groundwater treatment. And the reason we think for that is because groundwater contains not just nitrate, but it also contains silica. And again, all of these large diatoms, they all need silica to grow, to build their little crystalline shell that they use to protect themselves from viruses in the ocean. And if we go take a look at the chlorophyll results, so this top chlorophyll image is from the first experiment, and this bottom one is from the second. If you look at just skip to the bottom one for the sake of time, and see that actually the nitrate plus silica treatment grew the most. It showed the highest increase in chlorophyll, which was then followed by the groundwaters. So this really led us to believe, and I think this is something that's been documented actually in many different studies now that look at the response of phytoplankton to SGD, is that in many different ways. Some studies even just looked at cell size, which diatoms are much larger than other phytoplankton. That since SGD had a lot of nitrate, it was dominated by diatoms, since diatoms have the ability to break down nitrate, unlike dinoflagellates and many other phytoplankton. However, the diatoms were co-limited by nitrogen and silica. So just the nitrate treatment in the chlorophyll results of the second experiment came in much lower than the nitrate plus silica and the groundwater. Basically, and if we were to look at the nutrient results for this, which are in the full paper, you can see where this treatment actually ran out of silica by the end of the experiment. So the diatoms had to stop growing because they didn't have enough silica. And I think as a result, like, silica is understudied and underappreciated in its role in its ecosystem response to SGD because of this. If you're talking about phytoplankton and SGD, and specifically diatoms, quantifying silica, taking a close look at silica should be, I think, an important part of your study, especially since diatoms can break down that nitrate into other forms of nitrogen that then other phytoplankton like dinoflagellates can then use. Okay, the next study I want to talk about is looking at, it's another incubation, again in Alaska, and we're looking at many of the same phytoplankton genera that we're looking at in that study. This study got started because I actually published previously a paper that calculated the fluxes of nutrients to the Gulf of Alaska. And then after that paper felt like I did an incomplete job because I did not assess the biological impact of that. And I made a lot of claims on that paper, like, oh, it can produce so much, primary productivity based on the nitrogen flux, specifically comparing it to rivers, because southern Alaska has a lot of fresh flow to the ocean just from rivers and nutrient flow, and a lot of the blooms in Alaska, the Gulf of Alaska, were attributed to that. So it felt incomplete, so we went back and did an incubation experiment. So this one, we only had time to do one incubation experiment. So we fed the phytoplankton groundwater, river water, and those specific nutrient additions. And like I said, this built on the previous study where we just calculated the nutrient fluxes. And the thing that I think is unique, maybe overlooked at this study, and should be done more in future studies about SGD biology interactions, is the comparison of groundwater to river water. Because really, in a lot of SGD studies, we compare groundwater to rivers a lot. Like, just the amount of fresh water flow to the ocean, the amount of nutrients, etc. You see that all the time, and there are very few studies that actually compare the biological impacts of that. So we'll look in reverse order for this one, looking at the chlorophyll. You can see here again the nitrate plus silicate, same story, had the largest increase. This was followed by just the nitrate, and then the groundwater and river water is actually pretty similar in terms of the total chlorophyll response in the study. What was different had to do with which genera responded to the river treatments versus the groundwater treatments. I'm actually going to skip this slide. So looking just at this scatter plot, there's a lot going on here. So we're wondering, like, we see these differences in general response between groundwater and river water. What is causing that? And it seems to be the salinity. So this top plot is the total abundance of each of these three genera, so Pseudonychia, Catastris, or Leptocylindris. Again, Pseudonychia is harmful. These other two are not really considered harmful. What we saw was with increasing salinity, Pseudonychia increased in abundance and Catastris decreased in abundance relative to each other. So what this shows is that even if the chlorophyll response is the same, you can have a change based on the genera, like what phytoplankton are growing based on the salinity. And that may be one of the key findings in when we're comparing groundwater to river water, especially if in this area the groundwater is brackish or saline. So we see more groundwater, higher salinity, more Pseudonychia. I'm not the only person who has found this. There's also been some studies in Alabama that actually found something similar, not in an incubation experiment, in the field. And river water equals lower salinity and more Catastris, for example. So this has implications in Alaska for, like, there's a lot of subsistence farming in Alaska, or subsistence fishing, including a shellfish, which people can get domoic acid poisoning from. So that might be something we can tie in the importance of SGD beyond just increasing primary productivity. And I want to highlight a couple of other studies really fast that I think made a big difference. This was in Jamaica, and what they found was that although the nitrogen in groundwater, it's very nitrogen enriched groundwater in this bay, increased the amount of turtle grass. So this is turtle grass. The amount of turtle grass growth, the biomass of turtle grass in this area. Fish did not eat it. They basically refused to eat it. They would actually live in the SGD area and leave it to go feast on turtle grass elsewhere because the N to P ratio was not favorable in the SGD area. It was so skewed towards N in the SGD area. There's so much nitrogen that's actually unhealthy for the fish. So the fish would actually leave that area. So we see, okay, maybe these nutrients in SGD are increasing primary productivity, are increasing biomass in an area, but it's not working its way all the way up the ecosystem. So I thought that was a really interesting finding of this paper. This is out of Hawaii. This paper blew me away when it came out. Looking at the impact of submarine groundwater discharge on reefs. They did a lot in this paper. I'm just going to talk about like one little aspect that really changed the way that I looked at a lot of ways that I do my own research. And that is, they did a really good job of integrating these ecosystem indicators, such as diversity. So this says, like, looks at how many organisms are in an area, but also the relative abundance of organisms, and species richness, which is just the flat out number of different organisms you have in an area. All of these sites were SGD impacted. However, some of them had much lower nitrogen from SGD. That's the ones in the red box. Than the others. The other sites had a lot more nitrogen loading from SGD. And what you can notice what they found is that species richness was much higher in the sites that were much lower end, so like slightly less impacted. And then they also had more corals and more diversity of organisms. So like just 17, 13 different types of organisms versus 6 and 9. It's a lot higher. Also, the diversity was generally much higher in these sites. So diversity is important because you might have like 5 different species in an area, but 95% is just one species. That's lower diversity than if you have an equal amount of each of those different types of species. And they also looked at N15 in the different tissues, again, of ulva species, so of that sea lettuce, to link that this change was actually as a response of SGD. And you want to talk about impact and what's possible when we actually assess how biology is being impacted by SGD, is that there was a Supreme Court case that used data from this paper to actually win. So we have the Clean Water Act here in the US, which protects, says you can't just discharge pollutant water or effluent, sewage effluent straight to the ocean. Hawaii uses injection wells, and the Hawaiian Wildlife Fund went after the county of Hawaii saying, no, you're violating the Clean Water Act because you're polluting the ocean with all of this nitrogen. And Maui said, no, we're not, because we're not discharging it directly to the ocean. It's going through the injection wells. And through this data that really linked these changes in the ecosystem to the enhanced nitrogen from SGD, the county of Maui lost against this. So I think this is what's possible when we go look at actually assessing that impact. This is another fun paper. It's rather short, looking again at turtle grass. What they found again is that, yes, when there's more SGD in an area, more nitrogen loading, you get, here we have in the enriched, larger leaf areas, more dry weight. So basically the turtle grass is getting larger, but they put a lot less effort into reproduction. So the turtle grass that was exposed to SGD treatments in this case had much less flowering, which makes sense if you think about it. Marine plants, they don't have a brain, they don't have neurosystems, but they do have physiological responses to different stimuli. If there's a lot of nitrogen in an area, they want to stay there. They want to focus a lot on growing. If there's not a lot of nitrogen, they want their offspring, they want to reproduce offspring to go live elsewhere. So they put a lot more effort into flowering so that those new organisms can go move off into maybe a more favorable area. And I think this has implications for when we talk about managing coastal areas and coastal ecosystems, what it would be the better benefit. Would it be that you want to enhance sea grasses in a very small area, or are you looking to repopulate an entire bay? If you're looking to repopulate an entire bay, it's probably not good to have a concentrated SGD source in just one area. You probably want them to focus on reproduction and spreading those seagrasses elsewhere. This study looked at nematodes. So nematodes are very, very tiny marine worms or marine worm-like organisms. And what I thought was really interesting about this paper is the first time I saw a paper talk about opportunistic genera. So really, and this is something I've seen across a lot of SGD studies, when you have some sort of stressor from SGD, either you have a lot of nitrogen, some species take advantage of that, diversity goes down because that one species is able to grow a lot, or that one genera, or you have some sort of stressor like too much salinity or salinity going down, and a lot of other organisms can't compete. So like maybe one organism is better suited to lower salinity and then they survive. You can actually see a drop in diversity a lot as well. So I think this paper kind of changed the game in talking about that opportunistic genera, and that's something that we actually see a lot in SGD studies, even if it's not called that, and I would like to see that language integrated a little bit more. And this is the last one I want to talk about, just because I think this is very low-hanging fruit for a lot of people, for a lot of researchers out there, and that is just for this one subplot of this image that these researchers looked at. So they did a scatter plot of radon activity versus primary production. So primary productivity is more effort to calculate. I think you could easily just do this with chlorophyll to see if there are any correlations between radon, a tracer of submarine groundwater discharge. Higher radon activities means probably submarine groundwater discharge is near. To see if there is an impact. And you can even see here that they found a correlation between, one area where higher radon did indicate higher primary productivity, but not in other areas, which I think is a much more interesting finding than if okay, radon's increased, chlorophyll increased, end of story. We can then ask questions as to why did it increase in this one area and not in another? So that would be something that if you are looking to integrate more biological assessment into your studies, is to just do something like this, either with plotting radon or radium against chlorophyll concentrations. Okay, now we're going to go to the SGD biology database. So going back to your responses, I see lack of, what is limiting you from integrating more discussion in your area? Lack of prior studies in my area, hard to find too few studies, not enough knowledge about biology. I would like to help you with that, which does not necessarily involve directly me, but generally more for the SGD community. So I have created a database that I archive the information for all the biological studies that I see that actually assess the impact of SGD on biology, like actually measure the impacts on biology. And I've made it searchable so that you can find papers that are more relevant to you, whether it's what you're looking at, nitrogen enrichment, a specific type of fauna or flora, or a specific area. So if you would go to drlecher.net, and I'm going to switch my screen share. And then the database is right here on the upper right-hand corner. I'm hoping to eventually migrate this to its own website once it gets large enough. And on this initial page, I have listed them by year in which the study came out. So we have some already coming out in 2021. There's probably more that I need to add, going all the way back to 1967. So this is all of the studies that I've been able to find so far that actually assess that response. That's a lot to dig through. Let's say that you want to just know what the response is about fish. I did make this searchable. I try to use best practices with search techniques. The first type of database I've done like this. You can just put in fish and all of those studies will show up. Okay, there's been about 6 of them so far. And they do have links to the original paper. So like you can use that link to go to wherever it's sourced from. I didn't post these papers online. I just provided a link to the original source. Or let's say that you're working in a specific area, like Mexico. You can search Mexico and you can find various studies that have looked at the impact of SGD in Mexico, for example. Or you want to know the impacts of nitrogen on SGD. Brace yourself, this one's going to be a big one. I think there's like 3 pages here where you can take your pick of what do you want to know about in terms of SGD. Do you want coral reefs? Do you want, seagrass? What is it you're looking for? So I created this with the idea that it would make it easier for researchers to find studies relevant to them, especially because a lot of these studies about SGD and biological impacts have taken place in or have been published in other journals that we typically maybe don't read, like aquatic botany and aquatic biology. I think the typical SGD researchers is maybe not as familiar with those journals. So my goal here is to just try to keep finding those papers and make them more accessible to you all. If you have a paper that I have missed, or you know of one that I have missed, that actually documents a response, so does like one of these measures, please feel free to email me. And I'll put my email in the chat in case you need it. I would love to add it to this just so that it can get out there and people can see it and read it and find out more about your study and how SGD impacts marine biology. And then just to wrap up here, I've put in some suggested papers that you can like maybe take a screenshot of, or I'll stick around for a few minutes so you can take a screenshot that I would suggest if you're just getting started, you want to build that knowledge base to read. This one just came out this year, specifically on the microbiology of submarine groundwater discharge. So I'm pretty excited about that. I will say these don't go into they give a great overview. They don't go into as much specifics as you may need for your specific study site. That's why the database exists. I also want to provide you with a list of the papers I talked about for my talk today. So if you want to go read those and find out more about, which I would highly encourage you to do, the specifics of those really unique study designs as well. I'll leave that up. And then I also have one more, which is other papers that I did not have time to go over today, but I do think were really unique in either their findings or in their methods. So I'm happy to stick around and pull up various slides for you guys to take screenshots of if you would like to further your understanding of this topic by reading any of these papers. That's it. Thank you.
Thanks a lot, Alanna.